Professor Deepak Pental, who's very well known to us, not merely as Vice Chancellor of Delhi University, former Vice Chancellor, but a very distinguished scientist, Professor of Genetics now, and most unusually for a retired Vice Chancellor, actually very active as a professor and in research and so on. He's going to talk to us today about societal response to transgenic crops, apathy, ideology, or fear of the unknown, a subject of such immense excitement and importance and controversy in the recent past that I expect the hall will be filling up very rapidly because of this nature of the subject and the speaker. I hope we won't be too disturbed by people coming in at a steady pace. But I need hardly say that Professor Pental is the recipient of any number of distinguished awards, a whole series of them, as befits a person of his eminence and achievement. And what he's going to speak to us about is a subject on which he is utterly authoritative, and it is our great benefit to have a truly informed expert opinion and discussion on a subject that has provoked so much controversy. So, may I ask Professor Pental to go ahead.
Thank you very much for giving me an opportunity to speak here, because I think I have myself been beneficiary of listening to many interesting talks in this hall. I'll get down to my talk. The last century was really the most happening century in terms of developments in science and technology. In 1900, we had probably very little of what we take for granted today by the end of the 20th century. Aerospace, electronic goods, many developments in medicine also. The population in 1900, global population was around 1.6, 1.7 billion. By 2000, it was around 6 billion. This huge increase in population in that century was due to improvement in life standards and a great amount of research which was done in medicine, drugs, vaccines, diagnostics, so many developments. And we are all in the society quite well aware of the progress which was made in the field of medicine. We are very well aware of what happened in aerospace. We all know what happened in electronic goods. But we know very little in general about how this increased population was fed. How did the globe accomplish the task of feeding 6 billion people by the end of the last century, and today around 7.1 to 7.2 billion people. We all know that there is still hunger around, there is malnutrition, particularly in the developing countries. There is a scourge of obesity and overeating in the urban populations, and in the developed countries this is a major issue and leading to lifestyle diseases. So we are more aware of health-related issues, and that's understandable because from birth to death, human beings deal with health-related issues. But food somehow is taken for granted that, we have very little awareness of what big breakthroughs were made on the food front to feed this increased number of people. I think rural prosperity is a very important issue in India. Around 47% of our population is still dependent on agriculture, They may be doing some side jobs, but their main livelihood comes from agriculture. And as we go along, in another 20-30 years, it is projected that 42% people will be still on agriculture. So prosperity of rural India is of great importance and should be of great importance in our economic planning. And if we want rural prosperity, these are some of the points which we will have to look after. We need connectivity, we need more storage space, we need efficient supply chains. A lot of food is going waste. Reduced water usage is going to be very, very important, because water is required for industrial growth, water is required for improving the lifestyle of people. And therefore, water usage is going to be the most critical issue. Water, energy, and agriculture. I will rate it like that. Improved soil health. There is no doubt that due to lack of crop rotation, we are dealing with deterioration in soil health, micronutrient deficiency in soil. The carbon content of organic component in the soils is going low, and this is a very is going to be of critical importance that we find some remedies for these problems. Crop rotation, if you read even a high school book on science, people talk about crop rotation, but we are stuck now, let's say in western parts of the country, high yielding areas, in rice wheat rotation. In south we have rice. So crop rotation has gone out of the window. And of course, plant breeding, and with a goal of low input, high output. And I, as a student of science, can tell you that this is not a wild dream. It is a possibility, provided there is a proper policy framework to sort this thing out. Now, how did we beat the Malthusian predictions? Of course, Malthus made all these predictions in around 1800. At that time, one could see the shortages because the fertilizers were not even available. This is all the fertilizers, nitrogenous fertilizers. Haber-Bosch process came at the turn of the last century. Otherwise, we would have starved. Mechanization. Just imagine if we did not have agriculture based on fossil fuels, tractors and other farm machinery, how much of cattle, bullocks would be required to till the land, and how much you will have to grow as fodder for them to feed those animals. Plant breeding, after 1900, plant breeding became a systematic inquiry rather than a hit and miss thing, because by 1900 we had found out the Mendelian laws of inheritance, and systematic breeding actually started after 1900. So all these things, fertilizers, mechanization, plant breeding, are basically the last century's blessings to us, along with the medical things and along with other inventions which improve the quality of life. And of course, management, without managing your affairs well, without proper policy framework, nothing. Science and technology is also helpless if your policy framework is not good and management is not right. So, this is a slide which is based on great Russian geneticist work, Vavilov, who gave the theory of centers of diversity for agriculture plants. Human evolution may go into thousands of years, but it was only 10,000 years ago that plants were domesticated in the Fertile Crescent in the Israel-Jordan area, and the first domestication took place and first sedentary human settlements took place. Before that, human beings were mostly hunter-gatherer societies. So this shows you different parts of the world. The oldest is this barley, wheat in the Fertile Crescent. Even cattle was domesticated in this area. And this was the first area. Jared Diamond has a brilliant book on domestication of crop plants, better than many of the plant biologists have written. And I found that book which can be read by almost everybody to gain from how germs spread and how sedentary life started and human civilization started as we know today. Since then, a huge amount of genetic changes have occurred. Corn, of course, is about 5,000 year old domesticated crop. This is the wild relative. Teosinte is the wild relative of corn. And you cannot make much out of this. It might have been consumed by human beings, hunter-gatherers, but you will see that it is an enclosed grain, very difficult to crush to get the grain out. And this is the wild relative of our corn plant, and this transformation occurred about 6,000, 7,000 years back. And these are the first cobs. Today you must be eating maize cob sometimes. You see the size of that and you see the size of this cob. Some of the archaeological work, the maize cob size 7,000 years ago and 500 years ago. So human beings were continuously selecting for bigger cob size, accumulating the genes. Of course, the genetic basis we know now, but you can do, it's like selection in dogs, which was done without any great knowledge of genetics, but people bred dogs by selecting and preferential mating. And in around 1930s, some of the very brilliant scientific work on plants, people showed that hybrids are higher yielding than the maize varieties that were being grown. And you can see that hybrid corn was introduced in around 1935, and by 1960s, 98% of the area in the United States was under this corn hybrids which are made by human intervention. So what I want to convince you about is that from that wild relative about 9,000-10,000 years back to modern day corn, lot of genetic modification has been done, and this process got hastened in 1930s when hybrid corn was, and this crop has really all the beef industry and all the high protein intake which Americans and Western countries are having is based on the corn revolution. We were staring at famine in South Asia, and this is the first dwarf rice which was introduced by International Rice Research Institute around 1960, and how the area spread, and it was called miracle rice because those who remember those days, I was a very young person, most of the young people can only read about it today, but it was really a desperate situation in South Asia. Look at IR64. Now these are series of varieties that have been released by International Rice Research Institute. What complicated breeding programs. Sometimes we don't appreciate how much labor, because in appearance the plant will look very much the same. It has not changed much. The dwarfs look little different from the tall varieties. But otherwise in the breeding, you have to keep the plant looking the same, but increasing the yield. If you meddle with it too much, it may go haywire. So breeding in these complex pedigree programs is a very tough job, and maybe IR64 took about 20 years to breed in the breeding programs, and how much germplasm was taken to breed that. And a lot of genetic changes, recombinations, inversions, translocations happened during the breeding process. Just to give you another breakthrough was the dwarf wheat. This wheat genes came from Italy. From Italy to Japan, the Japanese breeder bred these Norin varieties, which were dwarf. When the Americans were occupying Japan after the Second World War, they really liked these dwarf wheats. So they took the dwarf wheats to breed for the United States. And their Ford Foundation had a program that you can get one nursery in Mexico and one in Pullman in Washington. It didn't start with philanthropic act, it started out of necessity, but they realized that Mexican population is increasing, there is a threat of communism coming to Latin America, and I am one of the believers of that theory, that they felt that we should intervene and do something for Mexico's food conditions too. And Borlaug is a bit of a pioneer because he left his family in Iowa and worked in very remote areas of Mexico and came up with the idea of shuttle breeding. But let there be no doubt, the dwarfing genes started from Italy, went to Japan, went to US. The US agriculture benefited tremendously, just as much we benefited. All varieties are now dwarf varieties. Now what is this genetic engineering all about? This is also a new dimension in genetic modification. What we have been doing, it adds another new dimension. I am one of the believers that we should not say that it is continuum. It's a bit of a break. It's like turboprop and jet engine. There is a quantum leap. It's like transistor and chip. So there is a breakthrough, and this breakthrough came through some very solid research work which was done all over the world, particularly in European countries and in the United States. Some work was done in India, mostly on cell culture side, mostly on tissue culture, cell culture side. In fact, Delhi University's botany department was one of the pioneers in some of the techniques. But we lost out on genetic engineering. We didn't have the money, we didn't have the trained manpower. Basically, it was the United States which was the leader in genetic engineering, whether you take bacteria or animal cells or plant cells. And of course, European science has always been very good. So I have tried to explain in a simple way that so far the genetic modification was from the primary gene pool, land races and varieties by sexual hybridization. We moved and did very solid work on utilizing the secondary gene pool, which is the near relatives of wheat, rice, soybean and so on, to bring genes from chimpanzee into human beings, if I can take a crude example. And now we are in a position to take genes from anywhere, from any organism, and to put it into plants. Nowhere, like the BT cotton, which is so much talked about, we were using BT preparations to spray it on high value crops in California and in some European countries for many, many years. We were dusting them, dusting the bacteria. But it was not possible that any bacterial gene could have been put into a plant. So this new gene pool which has become available is certainly a major breakthrough. And the point is how to use this major breakthrough for the good, for rural prosperity and for increasing the pace of plant breeding, which has been too slow. And let me add one more thing. Lot of low-hanging fruit have been already captured by conventional plant breeding. Now you are dealing with far more complicated situations where in many cases conventional plant breeding does not have the right answer or cannot do the job for you. So it was very timely that these things were found. I don't want to bother you too much. This is a very busy slide. I picked it from one of the top journals which is on plant sciences, Plant Cell. And you see the developments which happened over the years in cell and tissue culture. We talk about stem cells today. In plants, in the 60s and 70s, we could have a complete plant from a single cell. And that means if you alter the genetic composition of that single cell, you can make a plant with that change. And I have been a student of this subject for almost all my life. So I have seen this fabulous science unfolding from my undergraduate years to when I'm near retirement. So all I want to say is that there has been brilliant science in which millions of dollars were spent. We contributed very little to it, but we and other parts of the world can benefit from these fundamental discoveries in how to put a gene into a plant cell and how to regenerate a complete plant out of it. I would not have the time to go through this fascinating story, but let this be very clear that some wonderful science and technology development has gone into the area of genetic engineering, whether it is microbes, whether it is animal cells, or it is plant cells. What's the situation today? These green parts are parts of the world where transgenic crops are being grown. The graph is going up and up. It could have been more if there was not so much opposition in parts of the world and societal misgivings on the technology. It would have been double or triple than what it is. Even then, you can see that around 180 million hectares of land in 2013 was under transgenic crops with about 4 or 5 characters. And economic analysis, and I'm not going to go into economic analysis at all, but some of the peer-reviewed papers, I'm not talking about hearsay, where people have published papers in PNAS or some of the leading journals, they have shown that the developing countries have benefited more wherever they have introduced transgenic crops than the developed countries. This is with the controversial BT cotton, which now covers almost more than 90% of the cotton area. The cotton production has gone up, as you can see, and the acreage under cotton has also increased from almost from 7 million hectares, we have moved to more than 10 million hectares. How did it happen? We were importing cotton at one time, now we are exporting cotton. People say that hybrids have also helped the productivity. There's no doubt that hybrids have also helped productivity. I'm not contesting that at all. But which farmer will buy hybrid seed if the crop is not protected from the insect? If everything is going to be eaten up, nobody is willing to commit more money into hybrids and more productive material. So another dramatic difference that transgenic technologies made is papaya ring spot virus. Entire Hawaii papaya cultivation was under threat. There is an antisense technology which was used, and today I think all the papayas we eat have this gene in it because it has spread all over the world. There is hardly any part of the world which is not growing ring spot resistant papaya. At least in Taiwan, China, all over the place it is being grown. Every crop has a number of difficulties and problems. Like human beings have number of issues with diseases, same way each crop has a number of issues with difficulties. They could be insect pests, they could be fungal diseases, they could be bacterial diseases, viral diseases, and we have this continuous desire to increase the productivity of our crops. There's nothing wrong with aspiring for more productivity as long as your input doesn't increase. If with the same amount of fertilizer you can get more crop, I think that is an ambition which is worth pursuing, which is very important ambition, and every plant scientist should have it. Now, my main experiences with mustard, when I returned to India, I thought I should work with an orphan crop. on which nobody else is working. This crop is grown in about 6 to 7 million hectares of land, mostly in the dryland areas. This is not a crop of high input. This is a crop of very low input, mostly on residual moisture, but now irrigation has improved, and even in Rajasthan people are giving 1 or 2 irrigations to get more yield out of this crop. So there are a number of problems with it. There is an alternaria blight, a fungal disease. There is a major insect pest, aphid. There is another fungal disease. We need to improve the quality, so on and so forth. So all crops, and I will show you some slides on that, have difficulties which we have to deal with continuously. And let there be no doubt, even with conventional breeding, if I bring resistance to blight, it is going to break down after 5 to 10 years. So I have to continuously search for new genes, new sequences, whether I do it through transgenic route or through through plant breeding route. I need more sequences, I need more genes, otherwise the pest will win over the crop and the crop will be decimated. And this is the way how we have fed 7.2 billion people. This is the procedure through which we are feeding all the population. I want to give you a test, what is that called, a story from my own work, because why go too far when the speaker can give his own work to demonstrate something. When we started mustard work, we made one very interesting observation, and that was that when you cross Indian mustard varieties with East European mustard varieties, we were dealing only with Indian gene pool, which became very narrow. But when we cross it to East European types which were grown in Ukraine and Soviet Union, the erstwhile Soviet Union, you get hybrid vigor, you get more yield. Just like what people discovered in corn in the 1930s, we discovered that in mustard in 80s, that we can make mustard hybrids which will increase the yield potential of the crop. Now how do you make hybrid seed? You have to make one line male sterile so that it is female fertile but male sterile. So the pollen will come from the other parent, and the seed you harvest from this line will be the hybrid seed. That hybrid seed can be given to the farmer, and farmer will earn more because the yield has gone better. So we wanted to make a male sterile line. And in 1990, 2 papers in the topmost journal of the world, Nature, 2 papers were published in a male sterility through genetic engineering. Brilliant papers, whether we want to use it or not, but to me those papers were brilliant use of science to develop a technology. And what did they show? They showed that if you express a ribonuclease gene, an RNase gene, in a small part of the plant where male gametes are developed, these cells will die, and because these cells feed the male tissue, the pollen will die, therefore you will get effective male sterile lines. So we said, this is not patented in India, we should try this technology to see whether it will work for our hybrids, to develop mustard hybrids. And we found on the way that this technology can be further improved. We improved it. We got US, Canadian, European patents for the improvement that we made. Those patents are still held in our lab's name. And we could develop a full system in 2002, which is still hasn't gone to the farmers because we are locked up in court cases, and we have this tremendous difficulty with whether we should deal with transgenics or not. These were some of the biosafety trials which were done based on the biosafety protocols that have been laid down in India, and I think they meet all the FAO and all the global standards. They are no less stringent than any standard that have been set by those countries which have released transgenics. I'm not talking about those countries which are not releasing transgenics. And absolute precautions, everything. And these trials were done by the Indian Council of Agriculture Research, not by us, by ICR so that a third party does the test and sees whether there is any value in these transgenics. And I'm giving you the 2010-11 data. The hybrids yield about 20 to 30% more than the best varieties that are in the field. This is 2010-11 trial, and this is 11-12 trial. These are large plot level trials, so therefore the data is quite accurate. And in the 11-12 we got about 30% increase. And this is the trial, the third spot trial in Sri Ganganagar, which was burnt by Rajasthan government 10 days before maturity because some NGOs reached Rajasthan government and they developed cold feet. I requested them that we should be allowed to complete. Right now nothing will happen because the crop is in the terminal stage. And the answer given to me was, if you can get your readings done before the assembly session, basically Rajasthan government didn't want this question to be raised in the assembly. I want to show you that this technology which we modified and used in mustard, that was 2011 I think, 2012. These three genes which have been used for male sterility and restoration have gone through very strict biosafety analysis in Australia, Canada, China, European Union, Japan, Korea, South Africa and the United States. Hybrids based on this technology are being grown in Australia, Canada and in the United States. Rest of the countries accept the products of these crops but don't grow them themselves. There's a difference. European Union is importing huge amount of transgenic crops into their countries, but they are not growing it. So I want to make it clear. But from biosafety point of view, they have cleared it long time back. So it's a political decision by Europe not to grow these crops. It's not a biosafety based decision. And in Canada, using this technology, from 13 million hectares rapeseed, which is a relative plant of mustard, from 13 million acres, the crop is now in 16 million acres, and the production has gone from 6.5 million to about 11-12 million tons. And Canada is a huge exporter of canola oil all over the world, which is shown here. Japan, which does not grow any transgenic, imported 2.3 million tons of rapeseed, transgenic rapeseed, meal and oil. Oil, of course, does not have transgenic protein. Let me also clear that. But meal has all these proteins in it. We also made in 2004 a very interesting transgenic. I'm not giving you anything on the conventional breeding program that we have. About 80% of our efforts are in conventional breeding. I'm only telling you the 20% efforts because transgenic is the controversial area. We have developed strains of mustard whose quality of oil, compare the last column with olive oil. And you will see that we have now an oil in mustard which is better than olive oil because olive oil doesn't have 18:3 fraction, which is very important for vegetarians, while mustard is maintaining its 18:3 property, but we have got oleic fraction very high. This is 2004. This is developed through a technology in which plant's gene has been put in reverse order. No gene from bacteria, no gene from anywhere else. Only the plant gene put in a reverse order and no marker gene. The marker gene has been eliminated through markers. This is 2004 and this is 2014, 10 years, and we don't know whether it will ever go to the market or not and benefit human nutritional value. And also, I mentioned to you each crop has number of problems. Take rice. For rice blast, you can do conventional breeding. Difficult, but you can do it from wild sources. Now when you transfer a gene from wild sources into a crop plant, the good gene drags along with it maybe 1,000 other genes, because it's very difficult to break tight linkages. This is called a problem of linkage drag, which plant breeders face day after day. By transgenic method, you can just pull out the gene even from the wild relative and just put it into your crop plant without bringing all that baggage of bad genes, which you then take 5 to 6 years to eliminate from that, and sometimes you cannot eliminate because the linkage is too tight. Sheath blight, there is no way. This is rating of plant breeders. What is the most important problem? And I did this survey in 2002. People say, we don't know for what we need to use transgenic technologies and what can be dealt with conventional breeding. This is well known. This is a lie that we don't know. We know it fully well. And this was a committee which was chaired by Dr. M.S. Swaminathan. So people know. If you want to fudge the issue, it's a different matter. Sheath blight has to go through transgenic route. There's nothing available in the germplasm which gives resistance to sheath blight. So if you want to save yourself from pesticides, either you use fungicides, which is a possibility, and the world agriculture is using huge amount of agrochemicals, which I'll show you in one of the... Leaf rust in wheat can be dealt through conventional breeding. Spot blotch can be. In wheat, lot of problems can be dealt through conventional breeding. So you can say, let's not go into transgenics in wheat. Maize, stem borer. You cannot deal with stem borer. You need a transgenic technology for stem borer. So, same way, there are two implications of this. One, that there are a number of issues. Number two, who will put it all together? Please bear it in mind, when I come to companies, I'll tell you, first companies were litigating against one another, and now companies are doing a lot of agreements to bring a number of genes into a plant, because somebody has access to one gene, another person has access to another gene. Organic, this is sometimes flashed, organic. It's a good idea. You should bring organic manure into soils to improve the carbon content. You should do everything to reduce your inorganic fertilizers and switch over to organic or to manure-based fertilizers. There's no doubt about that. But this is a meta-study which was published in Nature, and there's another study which did not go to Nature but to another journal, which clearly shows that if you want to grow wheat organically, certified organic, you will require 40% more land under wheat to fulfill the production requirements which are today, 40% more. With legumes it is less, because legumes fix their own nitrogen. But for cereals, 30 to 40% more land is required. And this data, if somebody is challenging this data, I don't know what to do. This is a meta-study of about 500 organic studies which were done all over the world. Now, coming to companies, this is another issue which bothers us a lot. Monopolies. And the American politics was run, Democrats were for breaking monopolies and Republicans were for allowing monopolies to happen. But now everybody is in the same boat, Democrats and Republicans both, because they know that for global usage of a technology, you require transnationals. And this shows how many small seed companies and small entrepreneurial companies Monsanto has gobbled up in the past 5 to 10 years. So this threat is for real. This is very evident. And I'll show you some data on this further. And cross-licensing. First they were litigating. My gene is better than yours. But then they realized that no, the best idea is to take gene from there, from here, and now we have third generation of transgenics with 4 to 5 stacked genes already. Seed companies, this is 2007 data. I think the situation has changed even further. The top 4 companies, Monsanto, DuPont, Syngenta, and Limagrain, control about 50% of the global seed market. This is a reality. We cannot wish it away. And this is going to increase because these companies have access to technologies. And under Bayh-Dole Act, everything that American universities discover is patentable and is passed on to these large companies. If you don't want transgenics for stem borer, for aphids, for sheath blight, you'll go to chemicals. And these are the companies, Bayer, Syngenta, BASF, Dow. Around these 10 companies control 90% of research on agrochemicals and agrochemicals. If we are still not using these agrochemicals, the new generation agrochemicals, because we do not have the money to pay for them. So we are going to old-fashioned organophosphates and all that. Some of them are really very bad for human health. It is recorded, but nobody is talking about the pesticide and fungicide pressure on people, on land, on human health. When some people die, there is some news, and otherwise we go to sleep. We will have to move to next generation agrochemicals if we don't want transgenics. There is a way, and that's why I think it's very urgent for the government, for the politicians and for the society to decide. If you don't want to put your money into biological control through transgenics, you need to put your money into agrochemicals to get to make more advanced molecules which are more benign to human health. Some data on India and China and Germany. You will be surprised why I'm putting Germany. I want to make a point, and that's why I brought in Germany. China already had second green revolution through hybrids. We had only one green revolution. China already has two. We may like it, we may not like it, but China had second green revolution through hybrid rice. Their average yields are superior to our yields. We produce from 157 million hectares less than what they produce from 112 million hectares. And this is not transgenic. Now they have put $3 billion into transgenic research, 1,800 crores over the next 10 years, because they want to repair some of the damage which they have caused by hyper-intensification. They want to repair that damage. And they also want to go for biological control. But you look at India producing, this is paddy data, not milled rice, so it is a little bit higher than what you read, 268 million tons, China 540 million tons. Look at vegetables. India 109, China 573. If India was producing 5 times more vegetable than we are producing today, or even 4 times, you can imagine what will be the nutritional status of our people. This data is amazing. Now you come to chemicals. Germany, the hotbed of environmental movement. Herbicides. India uses 6,705 tons, about 6.5 tons, 500 tons of, 6,500 tons of herbicides. Germany 16,000. On only 12 million hectares of land. And they are preaching us. Not the government of Germany. They are not preaching us. Their NGOs are preaching us. Fungicides. 13,000 in India, about 10,000 in Germany, in only 12 million hectares. Because they have found new generation agrochemicals which are more benign. And they're using them in a big way, reducing it. If you see their graph that has come down, they're using less fungicides, less herbicides, less pesticides. Now, you see a great difference in insecticides. But don't forget, India is a tropical country. Every insect has a fine day in Indian climate. While Germany is a cold country and it has more fungal and herbicide-related problems and very little problems from insect pests. So I just wanted to show you that data, that we should not be complacent. Now we have a big system of ICR. Sometimes Jawaharlal Nehru is criticized that he did not put enough emphasis on agriculture. This is wrong. We just did not develop enough technologies. We had nothing in our research programs. So we had to get Mexican wheats. Let's not exaggerate. People were working And science is like that. Dwarf wheat was not found by Americans. It's a myth. It was bred into fine varieties by the Japanese. So science can appear anywhere. Big breakthroughs don't see the color or the face of a person. They come where people are working hard, where the people are discovery oriented, where the people are technology oriented. Success falls in their lap. But here we have 99 institutes, 68 agriculture universities. Why I am showing this? What are these people going to do? What are they for if they are not going to do frontline research to meet India's need of rural prosperity and low input, high output agriculture? But I don't think we are doing enough. I want to also mention how scientists can go wrong. There is a very interesting story of how this fellow Lysenko, very smart guy, he ruined Russian genetics by suggesting to Stalin that plants can be taught to grow in adverse climates. So if you stretch his theory a little bit far, tropical bananas can be slowly taught to grow in Moscow in the cold climate. Probably he didn't go that far, but all his thinking was woolly, Lamarckian, and he was instrumental in influencing the government so much that one of the most distinguished geneticists the world has produced, Vavilov, who traveled all over the world, collected germplasm from centers of diversity, which I showed, he was exiled to Siberia and he died in exile. We all know what scientists did in Nazi Germany. And I want to tell you today, if you look at C. Subramaniam's memoirs, he clearly says that Indian scientists opposed the introduction of Mexican wheat. They said it is dangerous. It was his political acumen that we are in real trouble, that he overruled all these guys. So scientists can also say very funny things. And science by itself cannot deal with certainties and say, this will not happen. Any scientist worth his salt would never claim that my discovery and what I am saying is the final word. In fact, we are taught in science, any science, as you say, question. Please ask, can it be falsified? Can it be changed? Can it be improved? So, where is the opposition to transgenic crops coming from? One of the things I mentioned to begin with is that, people are away from this technology. And frankly, we are making very little effort in explaining people modern science through documentaries, through video programs and so on. We have a lot of tamashas, political tamashas being exhibited every day on the television, but very little on, science and technology, very little material. So how do we reach out to common person who has been even told that in transgenic food can contain cobra gene, it can carry scorpion gene, as though all scientists are nutcases and they are out to put scorpion gene or some weirdo things like that. NGOs receive a lot of money. I heard somewhere that every year about 100,000 crores comes to our NGOs. Some of them are very good people. Some of them we respect a lot because they worked for RTIs and some of the social changes that have occurred in the country. But some of them are completely motivated and are paid organs of what their masters say abroad. Ideological leanings, even scientists, as I said, suffer from that. Left, and I have a lot of friends in left, I respect left philosophy as far as fraternity and equality is concerned, but I have deep problems with their thinking. Always contrarians to, maybe I'm stretching the point too much, but Green Revolution was opposed because that utopian political formulation will not come through if rural side, if famines are not there. How do you bring about that ultimate revolution if there is social dissension? If everybody is eating well, probably there is no room for that. So maybe it is long-term frustration or what it is, but a lot of people criticize Green Revolution. Grandstanding, this is typical our phenomenon, or maybe it happens all over the world, but it is getting exaggerated here. What the ruling party has to say, opposition, whosoever is in the opposition has to oppose everything that is being said. So even on vital policy issues related to nation, there is grandstanding. Within ministries there is grandstanding. Who is smarter? And no desire to die. It's like a cricket team where the batsman is only playing for his average rather than for the team winning through. I see it, and I think this grandstanding is one of the big problems, why we are stuck in the courts and why government has not been able to. So quickly I'll go through this. This is what GM Watch and others say. GM foods won't solve the food crisis. If GM technologies are used properly, they will. GM crops do not increase yield potential. I showed you an example where for hybrid technology we can use GM and increase the yield without a grain of more input, either in water or in fertilizer. So transgenic technologies can be used in many, many imaginative ways for yield protection, and yield protection, like we say with electricity, every unit saved is a unit produced. Every pound of food which you save is adding to the production. So there's no difference between increasing the yield and saving the yield. In fact, it is criminal to make people sow and then suffer the fate of their crops being lost to pests and pathogens and so on. So yield must be protected, whether in storage or in the field. GM crops increase pesticide use. Now that is, I don't know who is saying that, and there's no logic in this. There are better ways to feed the world. Yes, if we all go vegetarian, if we all eat very frugal, and if we are willing to share the food, and if we promise that we will not make ethanol and other energy things out of it, maybe there is enough food. I agree with that. Other farm technologies are more successful. I don't know what they are. Crop rotation we have to bring in. We have to go more organic as much as we can, and there is no, in my belief, there is no contradiction between organic and transgenic. You want to improve your soil health through organic manure, who is stopping? It should be done. People don't, GM foods have not been shown to be safe to eat. Now, this can be controversial, and this has to be looked at. We are allergenic to a lot of foods which are already there. And we don't want to add to the food chain anything which is allergenic or toxic to non-target insects. That should be the bottom line of safety. That nothing allergenic or which is toxic to non-target organisms should be allowed to go through. I am all for it, and I think there are mechanisms by which this can be ascertained. If you are not happy with one report, go to the next one. If you are not happy with that, use the latest technology, send it to even Finland or Norway or wherever. But if there is a desire to sort this issue out, you can. And GM and non-GM cannot coexist. Yes, I agree. You cannot label. In India, a lot of food is sold loose. So this is a nightmare situation. It will increase the cost of food. We don't want to increase the cost of food. So once you release a GM, non-GM will be very difficult to keep separate. Unless you make zones and take some precautions. It can be done, but don't set an alarm bells running if there is some mixing up of 0.1 to 1%. That is possible. And we can't trust GM companies. If you don't trust GM companies, do the research in the public domain and do more knowledge generation in open source rather than depending upon the companies. And the way we are de-skilling ourselves through this nightmare of court cases and so on, I don't think anybody will be left in the public domain who would like to work with transgenics and do any research in the area. So what we need to do, we need to move from this kind of hyperbole of saying people will go infertile and all that, the last thing, if you tell somebody you'll go infertile if you eat, nobody is going to like that. But this is all propaganda. This is not the way to go. I've just made some suggestions at the end that if we want to put transgenic crops, and I think we need them very badly, and we need to do world-class research in agriculture, for low input, high output agriculture. This should be a major national goal. We will have to follow the substantial equivalence principle, that food should not be allergenic, it should not be toxic, it should be just as good as what. Precautionary principle is not science. It doesn't mean anything. It is like saying too many accidents happen on the road, so don't get up and go out in the morning. This may be stretching it too far, but precautionary principle is not science. It's not measurable. Where do you stop? Strengthen RCGM. These are the two bodies, RCGM and GAC. RCGM in Ministry of Science and Technology, GAC in Ministry of Environment. SAC PM has made an excellent suggestion. They should have full-time chairpersons. And these departments are sluggish. They are dragging their feet. They are indulging in this, again, grandstanding. Who is superior? Who is more important? Rather than addressing the task, rather than going for the proper outcomes. So they should have full-time chairpersons. RCGM and GAC must meet regularly. Like, GAC, you don't like brinjal, move to the next. Move to the next. Dispense away with all of them. But why are you sitting over them, creating a scene of paralysis? Be decisive. Either way you say this is not good, we are not convinced with the biosafety data, we reject it. I think that is a far better situation than saying, no, we don't know what to do. We are just sitting with like a pigeon before a cat with eyes shut. And, this is very important. All the good countries do give a decision document, which is put in the public space. When you allow field trial, there should be a decision document. What things you considered, what scientific facts you looked at, why you believe it is safe enough at that isolation distance that has been prescribed. This should be all informed to people. And now that the fears have been heightened so much, there's no other way but to be very open and transparent with people. There's nothing to hide. If this is not safe, throw it in a bin. Get on with another job. And decision documents are very important in that, and put it on the website and give 1 month for people to put in their concerns, their views, and then answer them and satisfy them. And if any conflicts arise, learn to work together. Ministries of Agriculture, Science and Technology and Environment should jointly give the ruling. Let me say this. There is no way you can guard yourself against rent seeking and doing things wrongly. Okay? The only way this can be dealt with is wrong decision making is through openness and through inducting people into RCGM and GAC who are dead honest and whose focus is that nothing allergenic and toxic should slip through, so that we don't add any more burden of allergies to the food chain. So I have said enough. If there are any questions, any points, I'll be happy to respond. Thank you very much.